Welcome to Carmel College and welcome to the biology departments. This pathway 1, is going to focus on how medical drugs work, specifically how do painkillers work. Today's first lesson is going to look at amino acid and protein structure. You will need to understand the structure of amino acids and proteins to be able to understand the biology that underpins how painkillers work. Welcome to this first lesson in a series of lessons which is going to be looking at some of the A-level biology that you'll be studying next year at Carmel College. Today we're going to start by looking at the structure of amino acids and proteins. You've talked about amino acids and proteins at GCSE, but we're going to build upon that knowledge that you've got from GCSE and take it on towards the level we'll expect at A-level. Proteins are the most versatile of all biological molecules. You can see a phone screen on the right hand side of the screen. This phone screen shows the phone loaded with apps. As you know, there are millions of apps out there, and apps can perform a whole variety of different functions. There's literally an app for everything. It's the same with proteins. There is literally a protein that can perform any possible function you can think of in the human body. At GCSE, you probably just learnt that proteins are essential for growth and repair, but let's just have a look at some of the functions that they can actually have and that we will study at A-level. Proteins can be structural. They can make up your bone, your cartilage, your tendons. Collagen is a protein that does that. You have keratin, which is a protein that makes up your hair. Actin, a protein that's responsible for the structure of your muscles. Enzymes are proteins you will have heard of at GCSE. Amylase, pepsin, catalase, they're enzymes you might have heard of at GCSE. There are over 10,000 other enzyme proteins out there that can perform a whole range of reactions within your body. Transport as well, hemoglobin, that substance in your red blood cells that transports your oxygen, that's a protein. Motors, myosin is a protein found in your muscle. It acts as a motor to help contract your muscle. Hormones, insulin, glucagon, hormones that regulate your blood glucose levels. Those are proteins. Receptors, the reason we can see is because of the light that we're detecting coming into our eyes. It's a protein called rhodopsin, which acts to do this. We also have proteins performing roles within the immune system. For example, antibodies, they are proteins as well. You now need to make sure that you've got your worksheet alongside you, which you're going to be working through alongside this video. You will need to stop this video at various points to complete the activity before allowing the video to progress. You'll be aware from GCSE that proteins are made up from smaller molecules called amino acids. There are 20 different naturally occurring amino acids that exist. And different proteins have different combinations of these amino acids, which leads to them having different shapes and structures, which gives them the different functions that different proteins possess. At A-level, you will be expected to draw the general structure of an amino acid from memory. This general structure is shared by all 20 amino acids. Draw out the general structure of the amino acid on your provided worksheet and pause the video while you are doing so. You need to recognise that the amino acid is broken up into 3 key sections. We have the amine group, which is composed of a nitrogen atom bound to 2 hydrogen atoms. The carboxyl group, which is composed of carbon double bonded to an oxygen and bonded to a hydroxyl or OH group. And we have the R group of the amino acid. The R group of the amino acid is the bit that varies dependent on which of the 20 amino acids we are drawing. But from memory, you only need to be able to draw this basic general structure. So can you add those labels, please, to your amino acid diagram on your worksheet, and pause the video while you're doing that. The R group is sticking up from the central carbon. It's this bit, as I said before, that varies between each of the different 20 amino acids. There are 20 naturally occurring amino acids, and so there are 20 different R groups. The good news is that you don't need to learn how to draw all 20 different amino acids from memory. All you need to be able to remember is how to draw the general structure of the amino acid, which has come onto the screen now. In an exam, however, you might be asked to draw a specific example of an amino acid. In order to do that, the exam board would have to provide you with a table showing the R groups of the 20 different amino acids. Let's have a go at this particular example. The examiner in this situation wants you to be able to draw a molecule of alanine. Alanine is one of the 20 amino acids. The examiner expects that you are able now to draw the general structure of an amino acid from memory, and they will provide you with the table on the right showing the amino acid R groups. You will simply need to look for the particular R group for the particular amino acid that you've been asked to draw, and then replace the letter R with that group. In the case of alanine, the R group is CH3. We remove the R and we replace it with CH3. As simple as that. Have a go now on your worksheet. Pause the video at this point so you don't see the answers. And could you now try and draw out the amino acids glycine, cysteine and aspartic acid using your knowledge of the general structure of the amino acid and the table in the top right hand corner, which shows you the R groups of the different amino acids. As I said, please pause the video at this stage because the answers will be revealed on the next slide. Unfortunately, my writing is not the best. That's why I like to type things out. And I've been trying to draw on my screen on my tablet, which obviously makes it slightly harder again. These are the answers that you would have been looking for this particular example for glycine, cysteine and aspartic acid. You don't need to draw the general structure and the R groups in two different colours. I've just done that to try and make it easier for you to visualise. It's absolutely fine to draw the whole molecule in black or blue pen, whichever you require. As you know from GCSE, amino acids can be joined together to make larger molecules called proteins. We're going to learn how amino acids join together, and we're going to recognise that when two amino acids join together, we call the molecule a dipeptide. If we join many amino acids together in a big long chain, we form a protein molecule. A condensation reaction is the name given to the reaction which joins two amino acids together. The reaction always produces a water molecule and a covalent bond which links the two smaller molecules together to form one larger molecule. I would like you now to draw a circle around the parts of the molecule where the reaction will occur. This is where you see two hydrogens and one oxygen being close together. You should hopefully have identified that the hydrogen and the second hydrogen and oxygen are close together are on the carboxyl group of one amino acid and the amine group of the other amino acid. The hydrogen and the OH will be lost when the bond forms between these two molecules, and that will form a molecule of water, H2O. In a second, I'm going to ask you to draw the resulting products of the reaction between two amino acids when they undergo a condensation reaction. To help you to do this, I'm going to show you a quick brief animation on the next. Here you can see 2 amino acids at the top of the screen. The 2 amino acids are going to react in a condensation reaction to produce the dipeptide. You should circle the OH on the carboxyl end of one amino acid and the hydrogen on the amine group of the other amino acid. Those are going to be removed and will result in the formation of a water molecule, and a new bond will form between the carbon and the nitrogen, which is what we call a peptide bond. The newly formed molecule is a dipeptide, which consists of 2 amino acids joined together by a peptide bond, which is a covalent bond. This reaction, as we said, is a condensation reaction. Have a go on your worksheet now and see if you can draw out the structure of 2 amino acids and then the resulting products of the reaction. I do apologise for my handwriting, it is not the best, especially when I'm writing on my tablet screen. This is what your answer should look like, and you should have highlighted the peptide bond between the 2 amino acid molecules. Don't forget the water molecule. It's something that students always commonly forget to include. They draw the nice dipeptide molecule out, but they forget that water is equally important. It's one of the 2 key products of this reaction. Now I would like you to have a go at drawing the reaction between 2 specific amino acids. Remember I said before, you don't actually need to know how to draw from memory the structure of valine and methionine. You only need to know the general structure of amino acid. But in an exam, an examiner may provide you with the table that you saw earlier that showed the R groups of each of the individual amino acids. You can now use that table of R groups to help you to draw the structure of the amino acid valine and the amino acid methionine. Once you have done that, I'd like you to show me the product of the combination of those 2 amino acids, the dipeptide. And I'd like you to include any other products that are formed in this reaction. Pause the video at this stage because the answers will be available to you on the next slide. Here you can see the expected answers for this particular activity. You can see the amino acid valine, you can see the R group highlighted in blue, and you can see the amino acid methionine, the R group highlighted in blue. As we said, all amino acids have the same basic core general structure, the bit highlighted in black. I've used blue to show the particular R groups of those amino acids. The 2 amino acids combine in exactly the same way as we saw in the general example. The OH and H are lost to form water. As I said, don't forget the water molecule. And a peptide bond forms between the carbon and the nitrogen as shown. And that's the dipeptide we would expect to form between valine and methionine. The final part of today's lesson looks at protein structure. We're going to look much more at protein structure when we come on to looking at enzymes. Remember, enzymes are proteins, and understanding the structure of those enzymes is essential to understanding how enzymes function. We know that proteins are long chains of amino acids linked together by peptide bonds. And we can see that in the diagram in the bottom left-hand corner, which shows the primary protein structure, the blue circles representing the amino acids, those amino acids joined together by peptide bonds. The protein is not yet functional, however, and it needs to fold up in order to get a specific 3-dimensional shape. The first stage in that folding is what we call the secondary structure of the protein. This is where hydrogen bonds form between neighbouring amino acids in the long chain of amino acids, and they form structures called an alpha helix, which are coils within the protein chain, and folds within the protein chain called beta pleated sheets. The protein, however, needs to get even more folded. It needs to fold on top of this secondary structure to create a very specific 3-dimensional shape. And that's what happens when the tertiary structure forms. when we get additional folds on top of the coils of the alpha helix and the folds of the beta pleated sheets. That particular structure is held together by additional bonds, which include hydrogen bonds, but they also include disulfide bridges, which are very strong covalent bonds, and ionic bonds, which also form as well. This particular structure gives the protein its very specific three-dimensional shape. Now, in terms of enzymes, we know that enzymes have a pocket on them called an active site. The active site is the part of the enzyme molecule to which the substrate can fit into and bind. That specific shape is created by the folding into this tertiary structure that we see here on the diagram on the bottom right hand side of the screen. Let's look in more detail at the bonds that hold the tertiary structure of the protein in place. If we look at this diagram, we can see that there are small circles. These small circles vary in colour, and these are the amino acids. The reason they vary in colour is because there are different amino acids in this polypeptide or protein chain. Peptide bonds form between each of the amino acids, creating this long polypeptide chain with a start and an end. You'll remember that different amino acids possess different R groups. Cysteine is an example of amino acid that has an R group that contains sulfur. If we look at our polypeptide, our protein, we can see that there are two purple, two cysteine amino acids contained within this chain. The R groups of these two cysteine amino acids will form strong covalent bonds called disulfide bridges between them. These bonds are very strong and they are not easily broken. As you can see, the two cysteine amino acids are not immediately next to each other in the chain. There's some distance between them, but that has caused the chain to fold up in order to form the bond between the two sulfur-containing R groups of the two cysteine amino acids. So here we can see this folding, creating this specific three-dimensional shape. Different amino acids have different R groups with different properties. Some amino acids have positively charged R groups, while others have negatively charged R groups. We can see here that the orange amino acid has a positively charged R group. That represents the amino acid lysine. The grey amino acid has a negative charge. That is aspartate. We can see again that the chain is folding in order for the ionic bond to form between the positive and negative R group. We also see hydrogen bonds forming, which help to fold the chain further. They occur between R groups that have a slight positive and a slight negative charge. Because the charges are only slight, these bonds are only very weak, and they can be overcome by heating the protein, which will cause the molecule to vibrate and cause those bonds to break. That's what happens when the protein is denatured. Those weak hydrogen bonds break, the protein unfolds, and the shape of the protein is no longer suitable to its function. Finally, we have some R groups which are hydrophobic. These hydrophobic R groups want to embed themselves within the middle of the protein so that they're not facing and coming into contact with water that can come in contact with the outside of the protein. As we can see there are two amino acids called valine, the two red amino acids with these hydrophobic R groups, which have buried themselves within the protein molecule so as to not mix with any water that might surround the outside of the molecule. This table looks at the different bonds that you find present in the different levels of protein structure. The only bonds present in the primary structure are the peptide bonds, which hold the amino acids together between one amino acid and the next in the long chain of amino acids. As we said, the secondary structure, those peptide bonds remain between the amino acids, and we get the additional folding, either the coiling into an alpha helix or the folding into a beta pleated sheet. There's generally a combination of both of these within a protein, and it's hydrogen bonds, the weakest type of bond, which help to form these coils and these folds. Tertiary structure is folding on top of the primary and secondary structure to create this specific three-dimensional shape. So obviously we've still got the peptide and the hydrogen bonds. We'll get some additional hydrogen bonds, but we'll also start to see ionic bonds and disulfide bridges forming between the R groups of neighbouring amino acids. The strongest of all of the bonds present are the covalent bonds. Peptide bonds are a covalent bond, so too are the disulfide bridges, which, as we said, you only find present in the tertiary structure. Ionic bonds are the next strongest bond, but those bonds can, as we said, be disrupted by fluctuations in pH. So if the conditions become too acidic or too alkaline, they can actually disrupt and break those bonds. Hydrogen bonds can be overcome fairly easily, so by raising the temperature, and this is obviously, as we said, is the case with enzymes when they are denatured. The high temperatures break the hydrogen bonds. They change the shape of the protein molecule, which will change the shape of the active site on the molecule, meaning that the active site will no longer be complementary in shape to the substrate, meaning the substrate can't bind, meaning that the enzyme can no longer catalyze the reaction. And it's the hydrogen bonds which are broken when temperatures get high, because, as we said, they are the weakest type of bonds. Finally, we're going to look at the answers to the questions, the multiple choice questions on amino acids and proteins. The first question asks, what name is given to the part of the amino acid molecule that varies between one type of amino acid and the next? The answer to that is B, R group. What is the name of the bond that forms between two amino acids when they undergo a condensation reaction? The answer to that is A, peptide bonds. Glycosidic bond, out of interest, is the bond that forms when two glucose molecules join together to form what we call a disaccharide. We'll study that in the carbohydrates topic next year. Which of these bonds is hardest to break is question 3. It is peptide bonds. Remember, we said peptide bonds are covalent bonds. Covalent bonds are the strongest bonds, the bonds that are hardest to break. Ionic bonds are the next strongest bond. They can be overcome, though, by fluctuating pH levels. And hydrogen bonds are by far the weakest of the bonds. Question 4, which of these bonds is only present in the protein's tertiary structure? The answer to that is B, ionic. Peptide bonds are present in all levels of protein structure. They are the bonds that form in the primary structure between the amino acids. Hydrogen bonds are present from the secondary structure onwards, and they help to form the alpha helix structure and the beta pleated sheet folds. Question 5, what is the name given to the molecule form when two amino acids join together? It is a dipeptide molecule. Remember, a protein is when many amino acids, a long chain of amino acids, are joined together by peptide bonds. If it's just two amino acids joined together, it's a dipeptide. We can obviously add more amino acids to that dipeptide and turn it into a protein. Question 6, which molecule is always produced in a condensation reaction? The answer to that is water. Condensation reactions don't just happen between amino acids. They happen between different smaller molecules to form larger molecules, such as between glucose molecules to form disaccharides in carbohydrates. So dipeptide is one of the specific products if it's two amino acids joining together, but it's not always the case, because sometimes it will be other biological molecules such as glucose molecules that are being joined together, and when they are joined together they form a different type of molecule called a disaccharide. However, water will always be formed in any type of condensation reaction, whatever those small molecules are that are being joined together to produce larger molecules. I hope you've enjoyed today's review of amino acids and proteins. Keep safe, keep well, and we look forward to seeing you at Carmel College soon.